genetics timeline. In 1866, Gregor Mendel established basic rules of inheritance using around 10,000 pea plants. He established the three principles: the law of independent assortment, law of independent segregation, and the law of dominance. Mendel's laws represent a theory of particulate inheritance that describes how germ cells of most organisms transmit characteristics from one generation to the next. In 1882, Walther Fleming discovered the substance called chromatin within DNA. He noticed during cell division that the substance separates into thread-like strings, which later became known as chromosomes. In 1896, Friedrich Miescher was the first to identify DNA as a distinct molecule. He studied the composition of white blood cells at the University of Tübingen. He was able to collect amounts of pus from bandages to experiment and discover the isolated form of nuclein, which was a new molecule found in the nucleus of the pus cells. In 1910, Thomas Hunt Morgan established the chromosomal theory of heredity. While breeding millions of fruit flies, he noticed one fly had been born with white eyes instead of normal red color. He decided to breed this one fruit fly with one that had red eyes. In the first generation, all of three had red eyes, and then the other three had the white eyes. And in the second generation, the white eye trait dominated the red eye flies, and all flies that had white eyes were male. From this, he believed that it was possible for eye color and the sex of the fly to be closely related. He eventually deduced that the trait can be sex-linked, and also realized that genes proximate to one another tend to remain together. In 1928, Frederick Griffith worked with two strains of Pneumococcus pneumonia, a virulent strain with a smooth glycoprotein coat that killed mice when injected into them, and a non-virulent strain that did not. He discovered this when he mixed a heat-killed S strain with a live R strain, and the mouse still died. And the tissue remaining in the mouse contained live, smooth black bacteria similar to the S strains. It was then concluded that the S bacteria could transform the R bacteria, and the nature of the transforming principle was still unknown. In 1941, George Beadle and Edward Tatum proved how genes direct the synthesis of enzymes that control metabolic processes. Using bread mold spores to study the products of metabolism, they cultured the mold spores with sugar, salts, and the vitamin biotin, and allowed the spore to replicate. Some of the spores were mutated and would not replicate without arginine, found in DNA gene sequences. From this, they concluded that genes indicate specific enzyme production. In 1943, Fred Sanger worked to identify free amino acids in insulin and was the first to gain a protein sequence. He was able to determine that genes on DNA have a code and the codes code for proteins. In 1944, Oswald Avery, McLean McCarty, and Colin McLeod identified deoxyribose nucleic acid as the transforming principle responsible for specific characteristics in bacteria. They used mice and injected them with a lethal and non-lethal strain of bacteria that caused pneumonia. Mice eventually died, and the reason being was that the immune system could detect and destroy the rough outer coat of the innocuous R form of the bacteria, but not the S smooth form. From this, it was deduced that the R, when combined with the S bacteria, became deadly. And from there, the three purified the bacteria to obtain the transforming factor, to which they later found it was nucleic acid, or DNA. In 1950, Erwin Chargaff determined which nucleotide bases belong together. After studying a variety of species and the pairs present, he concluded that the amount of adenine roughly equaled the amount of thymine, and the amount of guanine roughly equaled the amount of cytosine. Also in 1950, Alfred Hershey and Martha Chase discovered that phage DNA, and not protein, was a genetic material. Not many people during this time were focused on using bacteriophages. However, the two of them took them and radioactively labeled their DNA and proteins. And after a process called centrifugation, the radioactive labeled proteins no longer remained. However, the DNA did. This led to the conclusion that DNA was the genetic material. In 1951, Rosalind Franklin used X-ray crystallography to discover the double helix form of DNA that DNA takes. She met Maurice Wilkins at King's College in London, and they each had their own techniques for doing X-ray crystallography. Rosalind discovered the structure was a double helix before Wilkins did, and shared the information with Wilkins. And she also held a conference that Francis Crick attended. And she died in 1958, presumably because of ovarian cancer, and was not credited with discovering the structure of DNA. In 1953, James Watson, Francis Crick, and Maurice Wilkins were credited with discovering the chemical structure of deoxyribonucleic acid. Using the evidence from Franklin and Wilkins, Watson and Crick were able to identify the shape of DNA and prove it was a double helix. In 1962, Crick and Watson were awarded a Nobel Peace Prize in Physiology or Medicine, and they shared it with Maurice Wilkins, who worked with Rosalind Franklin using X-ray crystallography to prove the crucial evidence. In 1955, Joe Hing Cho discovered the normal number of chromosomes in a human. 50 years prior to him, it was believed that the human genome contained 48 chromosomes, and he used a newly discovered technique to separate chromosomes in a cell and was able to determine that a human has 46 chromosomes in pairs of 2, totaling 23 pairs. In 1957, Matthew Meslin and Franklin Stahl were able to prove the theory that when DNA replicates, it uses a concept called semi-conservative replication. They were especially interested in trying to devise a way to prove or disprove Watson's and Crick's model of semi-conservative replication. In 1961, Marshall Nirenberg cracked the genetic code. He knew that the amino acids were the building blocks of proteins, just not where the amino acids were coded from. He discovered that 3 base pairs in a row represents a codon, which codes for one of the 20 amino acids. And finally, in 1962, Fred Sanger worked with Francis Crick, John Kendrew, and Aaron Klug, and investigated how to sequence RNA because it was smaller to work with than DNA. Sanger proved that proteins were ordered molecules, and by analogy, the genes and the DNA that make up the proteins should have an order or sequence as well. The techniques were then applied to DNA, and the deoxy method was introduced and used.